So there are some physical properties that and facts about RNA that maybe you guys should get familiar with before I start my section. These RNA, they're actually not very stable. So if you just compare DNA and RNA, I forgot what was the numbers. Yeah, I have it right here. So usually the mRNA, they can survive for roughly 6.8 minutes. And if it actually have the cap and the tail, it can survive even longer. But I just remember like seeing this. It was something really crazy that if you have a DNA, it can survive up to 7 years, like just not being degraded or something. I mean, it might be false.
Is that what is stored in ambient temperature or what is stored in
Yeah, in vitro, just natural ambient 37 degrees Celsius.
How long does it
I mean, not 37 is human temperature.
How stable is it, for
Well, that's the thing. Like within the cell, you constantly have the nuclease that's trying to cleave these guys because there might be a virus just floating around, and they're trying to kill them, and trying to like basically break apart the certain RNAs so that it can actually function, get picked up by other proteins to perform its duties. So it's within the cellular level, it's actually a lot more complicated than that. Like it's really harder to measure in terms of time. Which is, I think that's how they got the 6.8 minutes. Is they actually took like samples every time to try to figure out like what is the average lifespan of an mRNA before it gets degraded, like naturally, before it starts messing up, like have something happen to it. But yeah, so within the RNA, it cannot hold as much information than DNA. And they are very vulnerable to degradation. Apparently I didn't pay attention to what was typed on the slide. And 98% of the genome do not code for protein. And this kind of shocked me a little bit, just this statement over here. Okay, so what was it, like 10% of the genome is transcribed within humans? I forgot, like 20 or 30, that's potentially, I forgot the numbers, but it's a lot. But out of them, only 98% of them are actually coding for proteins. So which kind of, I mean, I know a lot of them might be just some inserts, random inserts from like viruses. It might just be junk. But there is the huge possibility that they might not be junk, all right? So that's kind of what we are trying to figure out, and why we're trying to study the non-coding RNA aspect of it. So here's one idea that this is now deal more with our research. Is a typical like folding of RNA. We try to simulate the conditions in which if you're given like a piece of RNA, like what is the minimal energy or the most stable state that it can be to occur, okay? But here's the thing. RNA aren't they're very short, and you only have like 3 different types of base pairing. So they can actually form into different types of shapes. This is just for more of a personal opinion, and just from reading papers. Now this is more of a touchy subject that I'm talking about. That I feel RNAs aren't really that stable. I mean, that's a fact, but they can actually form into different structures. And our research is trying to figure out what are the possibilities in forming different structures. And then by using that, we can capture a specific characteristic of the piece of RNA. So let's say if you just give a child, right? I mean, you kind of saw me doing this. So tell a child, grab this string and start just playing around with it. All of a sudden, you have this what we call the hairpin loop. So here's the loop region, here's the stem region, okay? And just tell the child to repeatedly do this. So after a while, you're going to have this really conserved characteristic of this piece of RNA. I mean, this is kind of influenced by the human, but the idea is similar, that over here, you're going to have a base pair. Over here, you'll always have this loop structure. So what we try to do is use entropy to find first we find the base pair probabilities of every single nucleotide. And then try to figure out what is the possibility that it not base pair with anybody. And then we will capture it using what we call entropy, and Amir is actually one of the person that is an expert in this area. Well, why we use this characteristic is we can basically distinguish specific types of non-coding RNAs that separate it from the background, like much easier than other tools can. Okay, so if you grab a chunk of sequence randomly from the genome and versus an actual non-coding RNA, we can look at their value, their entropy value, and have it distinguish that, oh, because it's more likely that if you repeatedly fold this, it's going to have the same structure. Therefore, it must be a non-coding RNA. Versus if you have a random sequence, if you do the same thing, you're not going to have the same effect. Is that kind of clear or confusing? Yeah? Okay, cool. So here are some of the studies that I did. So what you're seeing over here, I mean, I'll bring it into it. You will see this. So what you're seeing over here, this is what we call the HFQ protein. It's within the prokaryotes. I don't know if some of you guys heard of it before. Yeah, these guys, one idea is these RNA are very sensitive. I mean, they can get degraded very easily, right? So oftentimes you need protection. They need protection in order to function. So these proteins will wrap themselves, wrap the RNA. And basically, before you have something like this, like this is a piece of RNA. All the bases are within the inside, okay? I mean, once in a while you have a couple bases that's actually sticking out. But they actually, because of these guys, what I think is proteins will be able to identify them and kind of cut it or do whatever. And that's what the HFQ does. And so now, what I wanted to show over here is, so you have a protein. Once it wraps it around, now the bases are sticking out. Okay? So now you have the capabilities of attacking the target sequence that it wants to regulate. So here's the idea. Like you have the complex of a bunch of, this is the RNA sequence. If you have a transcript that's going on, it can potentially block it by just attaching to it. So that's the kind of some of the mechanisms behind it. Okay.
How many RNAs going to 14?
How many RNAs?
I mean, for the non-coding are those regular
Families of
Families? Well, from what I know from the data mining that I did personally, I found roughly 40. I mean, I have the database if you want to take a look at it. Yeah, basically this is some getting a little bit too much detail, but I took the sequence from the genome, the NCBI database, and I grabbed all the non-coding RNAs that exist within prokaryotes. I read them one by one. I list them one by one. I mean, it wasn't that many. But I basically tried to do some data mining, but then I didn't know I got the name. I didn't know what it does, so I had to look up them one by one within Wikipedia, within literature, anywhere I can find, and try to distinguish whether or not some of the RNAs are what they are, like what type of function. Which is how I did the following study. So what I realized